Okay, guys, so today we're going to begin our review of the metabolism, which is the pinnacle of what we've been trying to get into. We've discussed the endocrine system, we have then discussed the blood system, and then after that, we did the lymphatic system, the immune system, innate immunity, and also adaptive immunity. Then we embarked on the respiratory system and then the digestive system, and we are inching closer and closer and closer, trying to get to the cell. So all of these things are happening system wise. Now we are finally getting into the cell to see that all of this oxygen that we have been trying to get into our system, this oxygen, once it gets to the cell, before we've been so worried about how oxygen makes its way to the cell and how carbon dioxide makes its way out of the cell. Now we finally got to the cell, and now we're going to enter inside the cell and see what oxygen does inside the cell. Not only that, we're also going to be talking about nutrients, these macronutrients and the micronutrients. What's the difference between a macronutrient and a micronutrient? Is a macronutrient is so big, that's why they call it macronutrient, and the micronutrient is so small and tiny, that's why they call it micronutrient? No, a macronutrient is something that you need to ingest in large amounts to survive, to sustain life, while micronutrients are also important. Macronutrients are not more important than micronutrients because they are macro and the micro are micro. No, both of them are important, but they have to be taken in the proper quantities. So we're going to be talking about our different types of nutrients. Is water? That's a very important question. It's on the exam actually. Is water a type of nutrition or no? Is water a form of nutrition or not? Yes, it is. Is water a form of macronutrition or micronutrition? Macro, because you need water in large amounts. Very good. That's good. So, and then I see somebody left water here. One time yogurt, another time water. Macronutrients. Very good. So we have carbohydrates. So the food that we intake that is based on hunger and appetite. Once the food has made its way into our bodily system, it has to be separated. There has to be discrimination. We have to discriminate where is carbohydrate, where are lipids, and where are proteins. Are nucleic acids a form of nutrition? That's another question that's on the exam. Are nucleic acids a form of nutrition? No, we do not. What's nutrition? It's something that we get ATP out of. Do we get ATP out of the metabolism of nucleic acids? No, we do use nucleic acids to replenish the supply of our DNA. We use it to make the nucleic acids that are part of the ATP, but we do not directly use the nucleic acids to generate ATP. Um, then we're going to talk about the lipids, how we take the macro structure of the lipid, and which is a triglyceride divided into a glyceride form and free fatty acids. And then how do we get ATP out of that? And then we're going to talk about all the metabolic, the biochemical pathways that we shuttle our breakdown products of the lipids. We shuttle the breakdown products of the lipids into glycerol and free fatty acids. Then we shuttle the free fatty acids into the beta oxidation pathway. And then we shuttle the glycerides into the gluconeogenesis pathway. Then we shuttle our glucose into the glycolytic pathway. And then we perform protein catabolism and we get ATP out of that. And in the end, we have breakdown products that will later be shuttled into the Krebs cycle and the electron transport chain to maximize the maximal amount of ATP to be generated. We get ATP in two different forms. We get ATP directly, so in the process of glycolysis. We literally get out ATP molecules. But there are other byproducts like hydrogen protons, like NADH. Those will later indirectly be converted into ATP. Remember how we talked about maltose being glucose and sucrose being glucose and fructose, and later on we'll take fructose and convert it into glucose. So the same thing is happening in metabolism. So we don't only get ATP directly, we do an exchange. We take hydrogen protons and indirectly convert them into ATPs. That's all recycling. There's a lot of recycling that's happening where we take NADH, convert it to ATP, and we take hydrogen protons. Thank you. And we convert it to ATP. So, this concept, metabolism, you can never master it unless you have a very good understanding of your digestive system. So, we said that in our digestive system, we have three compartments. The first compartment is the digestive system. The first compartment is the digestive system. That's compartment number one. Then the second compartment is going to be our circulatory system. Circulatory system. That's the blood. And the third, that's number two. That's compartment number two. And compartment number three is our tissue. And in the tissue is obviously made up of a lot of cells. That's why we put dots, dots, dots. These dots represent for us nucleus. So what happens is once we ingest the food enters. We have plus nutrition that has entered into our digestive system. Then we digest that food, we break it down, we metabolize it, and then we send it to compartment number two through absorption, and now it's circulating in our bloodstream. Then the bloodstream will deliver our food supply into the tissues, and the tissues will perform metabolism. So all the food that we plus, the food that we have gained, we're going to metabolize it, and then we're going to send out the waste. So now your tissue is going to send waste into the blood circulation, and then the blood circulation is going to send the waste to three different places. We're going to send minuses to three different places. What are the three different places we're going to send the minuses to? Obviously, the first one is the digestive system for defecation. We need to excrete or defecate digestive system waste that are coming from the tissue metabolism. In addition to the digestive system, where else do we send the toxics and the negatives? We send to the respiratory system, because one of our metabolic waste is obviously carbon dioxide. So we need to shunt that and expel it into the atmosphere. Otherwise, a buildup of carbon dioxide is going to increase the hydrogen proton concentration in the blood, and that's going to lead to acidosis. What is the third place we send our minus? We send our waste, and we send our toxics, and we send our byproducts. What else? Yes. Excellent. The urinary system or the renal system, so that we can send out all of the urea and all of the hydrogen protons in the kidney tubules. Excellent. So our inlet is one, the digestive system, but our outlet is actually three: the digestive system, the respiratory system, and also the urinary system. You also need to remember that this digestive system, the best way to understand it is to break it down into anatomy, which is structure, and physiology, which is function, and lastly, there's lab, I'm sorry, there's pathology, which is what went wrong. What went wrong with this? So our pathology will always be both. There will be pathologies related to the structure. There are structural pathologies, like congenital absence of the myenteric plexus. You know that happens. Imagine a patient that is born lacking that myenteric plexus. What is the myenteric plexus? The myenteric plexus is one of two computers. in addition to the mesenteric plexus. And both of them make up our enteric nervous system. We said that the enteric nervous system is not any less inferior than your brain and the spinal cord. The spinal cord totals about 100 million neurons. How many neurons is the enteric nervous system? About the same number, 100 million neurons. Yet people assume that the nervous system is only the brain and the spinal cord. No, it's the brain, the spinal cord, and the enteric nervous system. The enteric nervous system is so important because it regulates the movement and the secretions. The movements in your digestive system are divided into two types of movements. What are the two types of movements in your alimentary tract? So they call it the alimentary tract, beginning with the oral cavity, and then the pharynx, oral cavity, and then the pharynx, the pharynx, the pharynx, and then you have the esophagus, and then you have the stomach, and then you have your duodenum, and then you have your jejunum, ileum, and then you have your cecum, and then you have your colon, and then the transverse colon, and then the sigmoid colon, and then you have your rectum. The movement here, there are two types of movements. What are the two types of movements? When food enters the mouth and leaves through the rectum. And we said essentially that food that has not been absorbed, food that has been rejected by your intestine and makes its way out through the rectum, that is a type of food that has actually never been inside the body because it has been sequestered all the time in compartment 1. That food never made it to compartment 2, your blood circulation. That food never made it to compartment 3, your cells, your tissue compartment. So it's been out here. So failure of absorption is the admission ticket. Once you are absorbed, then you will enter the blood compartment, and the blood compartment will deliver you into the tissues. But if you have not been absorbed, you are here and you will leave. You are in the intestine, and then you will leave through the rectum. So you actually are essentially still outside. There's no difference between food that's outside and food that has never been absorbed. Both of them are just outside your body. So in the alimentary tract, you're actually outside. You have not entered. You are not inside the body until you have made it through compartment 2 and 3. What types of movements are found in our alimentary tract? We have two types of movements. The first movement is the peristalsis. Peristalsis. And then we have another type of movement known as the mixing movement. The mixing movement. At any time and any place in your alimentary tract, you have both kinds of movements happening. You have peristalsis. What did we resemble peristalsis to last time? Like a what? Like a band. Very good. It's exactly like a band. So it's like, in the ocean, the surfing, you have one wave and then you have another wave. So the waves come out like consecutively. So these bands of your peristalsis, they begin as a band and then they move down. And as they are moving down, they are mechanically, like you have a balloon full of water, and you are moving this tight band, and the band is pushing mechanically all the food. But the band itself is electrical stimulation. It's action potentials that are happening in what type of tissue? If you have what type of tissue? Smooth muscles. So that is not happening in a skeletal muscle. A skeletal muscle is not going to have peristalsis. Your skeletal muscles, your skeletal tissue does not participate in peristalsis. It does respond to electrical stimulation and nerves that innervate your skeletal tissue movement-wise. But you do not have your biceps having ripple effect. It is this because your blood vessels do have some element of peristalsis, but it is the smooth muscles that mediate that peristalsis movement. So you can rest assured that if you take a biopsy anywhere anywhere in your alimentary tract, the upper digestive tract or the lower digestive tract, you will always have smooth muscles because they are the ones who will respond perfectly to electrical stimulation and provide this peristalsis. So peristalsis is generating multiple bands one after the other, and the multiple bands, they move in something called the law of the gut. So the law of the gut says that the natural movement of food or the natural movement of nutrients is from the esophagus downward out through the rectum and the anus. That is the law of the gut. Does that mean there are no breaking of this law? Does that mean there are no violations? Obviously not. There is many times movement against this law. That's how you have GERD, gastroesophageal reflux. Sometimes you have diseases that work against this law, like achalasia. Remember, we talked about achalasia, which is severe constriction of your lower esophageal sphincter, and then the food is unable to pass. So this law cannot happen. So you can have violations of the law of the gut in two different ways. You can have violations in the natural way, like pathologies, or you can have other pathologies that propel the food backward. So we have peristalsis movement that pushes the food downward in the law of the gut direction. And we also have a separate type of movement known as the mixing movement, where food is not just moving down uniformly. While the food is moving down, it is also being mixed. So it's being mixed in the esophagus. It's being mixed severely more in your stomach, but that does not mean the stomach is the only place where you have mixing. So the mixing is happening with the highest capacity at your stomach because your stomach has many layers. It has the serosa on the outside. Then it has your muscularis layer. And then you have your submucosa. Then the mucosa. It has multiple layers. So it has the most maximal amount of mixing. But your intestines also have mixing. And yes, sorry, can I hear you? What is it? So the layers of the stomach, when we look at, maybe we have a model here. When we look at the stomach from the inner core, remember how we talked about the blood vessel. The blood vessel has on the outside the tunica externa, and then the tunica media, and then the tunica intima. Same thing with the stomach. So the stomach on the very outside, it has the serosa layer. That's the hardest layer. Then moving downward, you have the muscularis mucosa. Then you have the submucosa. Then you have the mucosa layer that is mostly intimate with the core. So if in your blood vessel you have the tunica intima, and they called it intima because it's mostly intimate with the blood. So the mucosa layer is the most intimate in the core. So that makes it intimate with, who's gonna help Eric? It's gonna be, what kind of liquid is it intimate with? So in my blood vessel, my tunica intima is mostly intimate with my blood. So what do you think the mucosa is intimate with? We said it was green. Yeah, chyme and bile, that's in the liver, that's in the intestine. So now we have not made our way yet to the intestine. We are at the stomach. So we have the burning acid. It's intimate mostly with chyme, the food that is being broken down, and also the acid. Very good. Any other questions? No other questions. Only one question. Okay. So moving on from the stomach. So we have a lot of mixing and we have a lot of peristalsis. In the intestine, we still have peristalsis. That's how food moves down, and we also have mixing. Once we get to the colon, we still going to have peristalsis, and we're going to have mixing. But now the colon, the large intestine, is mostly concerned with the reabsorption of water and the electrolytes. So if I take a sample, so let's divide the colon into three compartments. 1, 2, 3, and that's our rectum. Let's call it 4. If I take a sample from each compartment, if I take a sample of the chyme that's here, I like very much what your classmate said. The chyme. What's the difference between chyme and bolus? What's the difference between chyme and bolus? Yes. Excellent, excellent. So bolus is what you is the product of your hunger and appetite, is what you chose to ingest. Once it made its way, so digestion begins voluntarily. You're chewing, you're enjoying with your taste buds that food that you have chosen to get in. Once it made its way to the pharynx, then it's out of your control anymore. Now, from that point on, it's involuntary and is regulated by the parasympathetic nervous system. So now, once it got, once the bolus made its way into the stomach, where you have a lot of acid, and this hydrolytic acid is helping with the chemical digestion of the food, and you have this mixing movement with the peristalsis that is churning and chopping up the food. The product of the churning and the chopping and all of these hydrolytic enzymes and all of this pepsinogen and all of the acid that's in your stomach, that mushy type of food that just made its way out of the um, out of the stomach into the duodenum, that is chyme. So this chyme enters the intestine, and a lot of enzymes are being secreted. You have the lipase to digest fats. You have your different types of carbohydrates breaking enzymes, and you have your peptidases that bring down the polypeptides of the proteins. Then, when we have finished all of the digestion, we are now heading into the rectum. If I take a sample of the chyme here at location 1, then I took another sample of the chyme here at location 2. Then I took another sample of the chyme at location 3. And then another example of the chyme at location 4. What differences should I find? Are they the same? They're not the same. Very good. So we agree to that. They're not going to be the same. What difference will these samples have? Yes. The further down, the less nutrients. That's very good. But this happened here at the ileocecal valve. So here there is a sphincter and there is a valve. Just like how in our feet you have these varicose veins and you have valves that are one way. This is a one-way valve. The digestion has ended here. Remember, in our kidney tubule, we have filtration. After filtration, we have reabsorption. So once you make it through the ileocecal valve, so this is the cecum and this is the ileum. The ileum is the last part of your intestine, and the cecum is the first part of your colon, of your large intestine. And there's a valve in here, there's a sphincter, and the sphincter is a one-way sphincter. When there is high pressure, at the end of the intestine, pushing through at this junction between the end of the intestine and the beginning of the cecum, known as the ileocecal valve or the ileocecal junction. Food has entered here, so there is no more digestion. So, food wise, Roman, food wise, there is no difference because we have finished digestion. There is no nutrients. We have already absorbed. So, absorption was done here in the intestine. So, food wise, nutrition wise, 1, 2, 3, 4, they're actually the same. What the colon does, it reabsorbs water and reabsorbs nutrients. So that is the function of the colon. So I'm basically asking you, what is the function of the colon? And we said the function of the colon is to reabsorb water and reabsorb the electrolytes. So the farther I go down the colon, the less water that's there, the less electrolytes that's there. So that means at 1, I have liquid chyme. At 2, I have semi-liquid, mushy gel type. A little harder now because I absorbed a lot of water. Once you are at 3, you have more solid than liquid. And once you are 4 in the rectum, you have no water in there unless you have diarrhea. But it's complete reabsorption of water, and now 4 means it's solid. So 4 is completely 100% solid, and one is completely 100% liquid. And then 2 and 3 are different variations. The farther you go down, the more solid you are, and the backward you go, the more liquid that you get. And we said the law of the gut is to move down from the oral cavity, from the oral direction, down into the rectum. But that means they're not. It's not a perfect law. Sometimes you have retropulsion. Sometimes you have retropulsion that goes against the law. But that is the main law. So we have peristalsis and mixing. Now going back to my same question, where we have my enteric plexus, that the first computer, my enteric plexus, and the second computer, the first computer is the my enteric plexus, the second computer is the mesonar plexus or submucosal plexus, and both of them are the enteric nervous system that regulate for me different things. The my enteric plexus regulate the mixing and peristalsis, while the mesonar plexus regulate for me all the secretions, whether salivary secretion or mucus secretion or acid secretion or digestive secretion. Any type of secretion is regulated by this. Is so important. Which of the two computers is responsible for the movement of the alimentary tract? The peristalsis movement and the mixing movement. It is the myenteric plexus. What is the other computer, the mesonar plexus, responsible for, also known as the submucosal plexus, and the myenteric plexus, also known as the Orbach plexus. The mesonar plexus is responsible for the secretions. Now, some people are nobody is congenitally born with an absent mesonar plexus. So it is very important. The most important of both is the mesonar, because it's responsible for secretions. If you are born with no mesonar plexus, you never can secrete anything. But if you're born with the myenteric plexus or the Orbach plexus absent, you will have some difficulties. Difficulties in what? What difficulties will a patient who's born with congenital absence of the myenteric plexus have? Yes, Simon. Excellent. Difficulty getting food down, and especially difficulty getting food out. So these patients were found to have problems with defecation. So the food with the gravity can make its way. So gravity helps a lot. That's why it's better for you to eat on an upright position, and it makes no sense for somebody to eat laying down, because gravity helps propel the food in the law of the gut direction. It is the defecation that's the problem, because in defecation, you're not just dealing with a mechanical down gradient movement. You have two sphincters. You have an internal sphincter that is regulated with your parasympathetic nervous system, that is regulated with your myenteric plexus. And then external, you have an external voluntary sphincter that you regulate. So just because the internal sphincter opened and there is a lot of defecation, doesn't mean it's going to go out. You're not going to do number two on yourself. You have a secondary sphincter that helps you, and that is regulated by your cerebral cortex. The problem with the patient of absent or congenital absence of the myenteric plexus is that their external sphincter that is supposed to help them is actually hurting them, because they have no myenteric plexus to regulate it, so it will fight back. It will assume that this is not the right time to get the defecation material out. So now they cannot fight two sphincters. They cannot fight the internal sphincter and they cannot fight the external sphincter. So they always have a lot of problems with constipation and getting the food out. Which patients are usually more susceptible to the congenital absence of which of the two plexus? The Orbach myenteric plexus or the mesonar submucosal plexus? Yeah, the myenteric Orbach plexus. Very good. Very good, very good, very good, very good. Okay. So now the we are now we have now talked about how the food moves down, and then we have to emphasize some more about the secretions of our intestine. So in the intestine, we secrete enzymes on a discrimination basis. If you eat carbohydrates, the intestine is not going to secrete lipases and enzymes that digest lipids and fats. They will secrete the proper intestinal enzymes that break down carbohydrates. If you eat fats, then your intestine will send the enzymes such as lipase to break down the fat products, not just the carbohydrate. So I know you guys think the word discrimination, politically discrimination means something bad, but in science, we test in neurological examination, we test patient's ability to discriminate between the surfaces of this will tell that their sensation is proper. They are able to discriminate that they have been poked with a sharp object or a dull object. So discrimination here means that you're able to know the difference between the two stimuli, to the two inputs that are coming, that are feeding into your nervous system, into your computer. So the intestine will first have to discriminate by taking a chunk inwardly, taking a bleb, taking a vesicle inwardly of the food that's in its lumen. So if this is the lumen of the intestine and there is carbohydrate, C, 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 that means carbohydrate. Lipids, L, 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 L. Okay, proteins, P, P, P. So now if the intestine, how does the intestine know when to secrete carbohydrate enzymes, and when does the intestine know how to send protein peptidases, and how does the intestine know when to secrete lipase? It will endocytose. It will take a fragment of the carbohydrates and take it intercellularly into the villi, the microvilli, these crypts of Leberkuchen, crypts of Leberkuchen. They will now find out that's a carbohydrate, that's a C. Let me send the enzymes that break down carbohydrates. They will endocytose with a vesicle, with a bleb. They will endocytose proteins, and then they will tell that's a protein. They will endocytose lipids, and they will tell, oh, that's a lipid. I need to send lipase. So in order to digest, that's also happening in the oral cavity. In your oral cavity, amylase is sent with the same type of process. So there is endocytosis of carbohydrates, and then the computer that is found in your salivary gland will say, wow, we just ingested a carbohydrates. He just drank milk. He ate maltose. He ate starch. So now the output will be to secrete amylase that will break down the starch. But if you eat proteins and if you eat lipids, the endocytosis will happen in your oral cavity, and then the salivary gland, the computer will internalize and say, whoa, the structure does not resemble a carbohydrate. There is no alpha glycosidic bond. This is not a sugar polymer. That's a protein. I don't need to secrete amylase now. Oh, that's a lipid chunk. That's glycerol and free fatty acids. I do not need to secrete lipase. They do say that, some books say that not only amylase, definitely there is no dispute that amylase is firstly secreted in your oral cavity. So carbohydrates are so rapidly digested as fast as they enter your oral cavity. Some books say that lipase also is begins to be secreted in your oral cavity. Okay, and then there are we talked about how your brain regulates appetite and hunger. Hunger is a deficiency, a drop. There is a hypoproteinemia. There is a state of a drop in sugar, hypoglycemia, a drop in proteins, a drop in lipids. So there is an overall drop in ATP, especially if you are the type of person who burns that that energy. So the input sensory signal through the afferent neurons will feed into your hunger center, and then the response will be to stimulate you to go and take a meal. Then after you take that meal, then all of these processes will take place. You know the difference between hunger and appetite. So appetite is how you personalize the experience of fulfilling or feeding that hunger stimuli. Okay, what you choose to eat and how you choose to satisfy the drop in your. There are different types of ways you can supply a deficiency in fats. So different ways to supply a deficiency in proteins, different ways to supply a deficiency in lipids. And then the digestive enzymes will, in a generalized manner, break down the chyme, and then your intestine will ensure to churn, your stomach will ensure to churn and break down all of the food products. And these food products, when they are broken down, that is for the purpose of making the chyme, making the chyme with high surface area, so the enzymes of your intestine can easily digest it. So we modify the surface area. What is the job of the mixing process and the peristalsis? Is to increase the surface area of the chyme that we have taken in order to help maximize this counter current. Maximize. So there is like a counter, the same way we saw a counter current exchange in the vasorecta. There's also a counter current digestion, where there is a maximized surface area for the enzymes of the intestine. There are a lot of enzymes that are not just specialized in digestion. There are enzymes that feed back into the satiety reflex to tell that you have had enough food and that you need to stop. And um, there are also hunger enzymes. So let's now go into the slides, and let's go into each of the diagrams and see what topics we need to go over for nutrition. You guys can go and download that ZIP folder on Brightspace concerning the metabolism and nutrition. Okay. Really? Okay, I'll do that. I think I uploaded it. Okay, I'll do that. Yeah, but you don't need them. All you need is the study guide, and go back into that big folder to match like what's on the study. Try to solve the study guide using that big folder, or you can Google. It's up to you, or you can use the book. Just make sure that you fulfill that study guide, and you're gonna do excellent. The ZIP folder. Really? That's strange. Okay, so speak to me after class. Hmm? Okay, so the body weight is determined by the body's energy balance. So the weight is stable if the energy intake equals the output, and the we gain weight if our intake exceeds the output, and we lose weight if the output exceeds the intake. So the weight seems to have a stable homeostatic set point. It varies from one person to person, and the combination of hereditary and environmental influences, about 30% to 50% of variation in weight is hereditary, and environmental factors such as eating and exercise habits account for the rest of the variation. Okay, so what does that mean? So, our weight is a homeostatic variable. We talked about multiple homeostatic variables that must remain within the green, the U range. We talked about the temperature has to remain between 37.2 degrees Celsius up to 37.6 degrees Celsius. Above that is hyperthermia, below that is hypothermia. Also, we talked about the pH. The pH has to be within the range of 7.35 up to 7.45. Above that is alkalosis, and below that is the acidosis. This is happening also for every single electrolyte in our body. Calcium has to be within a certain range. Potassium. The sugar glycemic index has to be within a certain range. Below that is hypoglycemia. Above that is hyperglycemia. Within that range is euglycemia. So the concept of hypo and hyper, both of them representing for us stress. Hypo is a stress, hyper is a stress, U is the normal set range for our homeostatic variable. That happens also for the weight. But the weight is a little bit more complicated because it depends on many factors, such as the gender, such as the height of the subject. So they came with this very important scale known as the BMI, the body mass index. So we calculate the body mass index based on two units. It is your weight in kilograms squared on the denominator and your height in meters. And when you convert your height to meters divided by your weight that you have converted into kilograms and square that, the number that you get, if this number happened to be between 25 up to 29, that makes you overweight. If you are from 30, if the number is from 30 up to 34, that makes you obesity class 1. And then 35 up to 40, that makes you obesity class 2. And then 41 and above, that is morbid obesity, where the patient now looks for options like gastric bypass surgery or gastric sleeve, and some other patients go for gastric band. But here you really need medical intervention. You need medical intervention all along. Do not assume that overweight patients must not get a consultation or obesity class 1. These are all sparks of the fire. But the more you go down in this spectrum, the more injury that's there. So remember, we began with cells homeostasis, the cells being in a range of homeostasis. Then due to stimuli, stressful stimuli, we move on to cell injury, and then after cell injury, we will have cell death. And if the cell death was manageable, meaning that the mitochondria was alive, then we will go right back to a normal state of homeostasis, because the cell would die in a protective manner known as apoptosis. Otherwise, if the defect is severe, we move on to acute inflammation, and then after that we develop into chronic inflammation. So the stressful stimuli began here with being overweight, and then moving from overweight to obesity class 1. So now the injury has intensified. Then moving into obesity class 2, then the injury is really severe. Moving into morbid obesity, then now this is a life or death situation. And the more down the spectrum, body mass index 42, 43, 44, 45, 50 and above, the farther you go down, then this is very serious. That's playing with fire. And then the patient keeps on as you move up in the index. You would have a system-wise defect in the musculoskeletal. You'd have musculoskeletal defect. You would have nervous system defect. You would have atherosclerosis in the heart and in the blood vessels, and you would have every possible system, every possible bodily system will become under inflammation and stress the higher the body mass index. Don't forget, there is another side of the spectrum. So if you are from 20 up to 24, that is the healthy range. If you go down 19, then that is malnutrition. So malnutrition is not any less dangerous than obesity. Malnutrition, there's a lot of diseases that are happening in impoverished worlds, such as Kwashiorkor's disease. These diseases are actually even more dangerous than the diseases that come with obesity, because a drop in the protein supply would lead to edema, would lead to a dysfunction in the balance, in the osmotic balance between the extracellular fluid and the intracellular fluid in your body. So Kwashiorkor's disease is a disease that immediately leads to edema, because the number of the proteins that you need to be present to conserve the osmotic balance, 300 milliosmoles, will not be there due to the deficiency of the protein intake, and that would cause the water now to move from the body compartment into the extracellular fluid, leading to edema. So, and you must understand that obesity is mainly based on appetite, mainly based on resorting to food as a form of delight, as a form of pleasure. The food is also connected to the release of many happy hormones like dopamine. So many people, they resort to food for a sense of happiness, sense of pleasure. And there is also another factor, and that is a hereditary factor. Some patients are born with hypersecretion of enzymes that trigger the appetite, while other patients are born with a deficiency, hereditary genetic deficiency in the enzymes that break down fat. So the fat tissue is stored in the body. In which type of tissue? Known as the adipose tissue. Very good. I think that was one of the questions on the exam. So the fat tissue is regulated and conserved in the body in the form of adipose tissue. This adipose tissue, how does it interfere with water? So we have a balance. We have a balance between adipose tissue and water. The more adipose tissue you have, the less water. That's why there's an epidemiological difference between the structure, the anatomical structure of males and females. Males tend to have more water in their bodily anatomy than females because females tend to have a more bodily compartment of adipose tissue, more lipids. Okay, appetite. Control of weight involves several peptide hormones and regulatory pathways that control the short and long-term appetite. So short-term regulators of appetite. These mechanisms work over periods of minutes to hours. It makes one feel hungry and begin eating. The appetite makes one feel satiated and end the meal. So there is a balance. If you are hungry, an afferent input signal will go into your hunger center, and the output will be to stimulate the eating, to stimulate the cephalic phase. Thinking about, yes?
Yeah, my question. Isn't it kilograms over meters squared?
Yeah, sorry. Kilograms per meter squared. Very good. But that number has to be within those ranges. It includes the peptide hormones ghrelin that stimulates and is released from the larger curvature of the stomach, and the peptide Y and cholecystokinin and amylin. Amylin mainly is secreted when there is a lot of carbohydrates to be digested. Cholecystokinin, CCK, is secreted mainly when? When is CCK secreted mainly? That's another question that's on the exam. CCK is secreted mainly when there are carbs, or when there are lipids, or when there are proteins. Choose the right answer, A, B, or C. Cholecystokinin is secreted mainly whenever there is carbs, lipids, or proteins.
Carbs.
Jeopardy. Carbs. Very good. Try, but wrong answer.
Wow, you baited me. That's so crazy.
Cholecystokinin is released when? Obviously no carbs, because Ryan said it was wrong. It's not cholecystokinin. Cholecystokinin is not released when there is carbs. And we know that proteins are the easiest because they stimulate the release of something that begins with a P. What is it? Proteins are digested with P. Peptidases. So obviously the only remaining answer is cholecystokinin. Very good. Lipids, very good. CCK, yeah. Is referred to as the gut-brain peptides because they act as two-way signals between the GI tract and the brain. So the cholecystokinin and the amylin and the ghrelin and peptide gammas, they are all secreted by your digestive system, and they communicate with the central nervous system to regulate the peristalsis and the mixing and the secretion of all the other enzymes. Okay, so short-term regulators of appetite. Ghrelin. Ghrelin is secreted from the parietal cells. Ghrelin is secreted from the parietal cells, and the parietal cells are found in the fundus of the empty stomach. It produces the sensation of hunger. So they stimulate food intake. That's very important to remember that ghrelin stimulates food intake and stimulates the hypothalamus to secrete growth hormone releasing hormone. And it primes the body to take advantage of nutrients that are about to be absorbed. And ghrelin secretion ceases within an hour of eating. So, peptide gamma is secreted by the enteroendocrine cells of the ileum and the colon that can sense that food has arrived in the stomach. Its primary effect is to signal satiety and terminate eating. There's also CCK, cholecystokinin, secreted by the enteroendocrine cells in the duodenum and the jejunum. It stimulates the secretion of bile and pancreatic enzymes, stimulates the brain and the vagus nerve, along with peptide gamma, and cholecystokinin acts as a signal to stop eating. Amylin is secreted from the beta cells of the pancreas, and it produces satiety and inhibits stomach activity. Then these are short-term regulators of appetite. Ghrelin, CCK, peptide gamma, and amylin, these are short-term regulators of the appetite. There are, however, long-term regulators of appetite, such as leptin. So leptin is secreted by adipocytes in the adipose tissue throughout the body. So the level of leptin is proportionate to one's own fat stores. So it informs the brain on how much body fat we have. And leptin stimulates the sympathetic nerve fibers to secrete norepinephrine, which in turn stimulates fat breakdown or lipolysis. Most obese people have normal levels of leptin, but some have a defective leptin receptor. So a lot of obesity will cause a defect in the leptin receptor. And we saw that happening also where? We saw that happening with our this is also happening in your endothelium cells. Endothelial cells have LDL receptors. They found a correlation between obesity and a defect in the LDL receptor. So the LDL receptor with people who are morbidly obese is no longer working. So what happens is a lot of LDL molecules. What are LDL molecules? LDL molecules are low density lipoproteins, and the reason why they are low density because they have more fat than protein. If they have more protein than fat, they will be high density. So these low density lipoproteins must be trapped by this receptor and endocytosed into the tissue in order to get out of the fat, all of the fat and all of the lipid, and break it down into glycerol and free fatty acids and shuttle it into our metabolic beta oxidation pathways to get ATP. But what happened is there is a mysterious reason where in obese people with morbid obesity, that LDL receptor is defective. So you have a lot of LDLs that are swimming in the bloodstream, but they cannot be endocytosed because the receptor that takes them in is defective. So what does that lead to? It leads to a lot of LDLs, and the LDLs will now start to interact with each other, make a meshwork, and they make a huge bolus, and then they start aggregating, and they start to accumulate at the corners of blood vessels, and they make atheromas or atherosclerotic plaques. And these atherosclerotic plaques begin to make like very hardened arteries, and then blood cannot pass through, and that leads to ischemias, and that leads to a deficiency in blood supply. Okay, so that is the relationship between LDL receptors and obesity. The same thing with leptin. So the more adipose tissue that's out there, the more defect will be in those leptin receptors, and now this fat tissue will not be stored, in the adipose tissue, and it will be all over the blood, leading to them clumping together and causing atherosclerotic plaques. Insulin. Insulin is secreted by pancreatic beta cells. It stimulates glucose and amino acid uptake. It also promotes glycogen and fat synthesis. So insulin is an anabolic hormone. It has receptors in the brain and functions like leptin as an index of the body fat stores. So it has weaker effect on appetite than leptin. So arcuate nucleus of the hypothalamus is an important center for appetite regulation. It has receptors for all 5 chemical signals that were just described, and 2 neural networks involved in hunger. So one group in the arcuate nucleus secretes neuropeptide gamma, an appetite stimulant. So ghrelin stimulates neuropeptide gamma secretion, as well as insulin and peptide gamma, and leptin inhibited. So insulin, peptide gamma, and leptin inhibit the secretion of, they inhibit appetite, while ghrelin and neuropeptide gamma stimulate appetite. Other groups secrete melanocortin, which inhibits eating. So leptin stimulates melanocortin secretion. And remember, other groups secrete melanocortin, and melanocortin inhibits the eating. So leptin stimulates melanocortin secretion and inhibits secretion of the endocannabinoids, which are appetite stimulants. So leptin is very important to shut down this and initiate satiety. Leptin is important to initiate satiety and inhibit appetite. Leptin is important because plus it initiates satiety. You are full. You're not supposed to eat no more. And minus, it inhibits appetite. So on obese people, what do you think is happening? Do you think people who are suffering from morbid obesity are more likely to have hypersecretion of leptin, or do you think they're more likely to have a deficiency in the secretion of leptin? A deficiency. That's a very important point. So obesity causes a defect in the receptors and the synthesis of leptin. And we know that leptin promotes satiety, stopping the food, and it inhibits, puts brakes on appetite, so you're not supposed to eat no more, and then it's gonna stimulate the satiety so you feel the sensation of being full. That's why obesity causes a defect in leptin secretion, and that's why patients who are morbidly obese end up with the opposite. They end up with a depression in satiety and a stimulation of appetite. Okay, so the principal pathways of appetite regulation by the gut-brain peptides. So we know that ghrelin has a positive. So whenever there is red, that's a brake, that's minus, that's inhibition. And whenever there is green, that's gas, that's stimulation, that's pressing on the gas. So ghrelin and leptin are the only 2 enzymes that are stimulatory, that are secreted by your gut-brain system, that are stimulatory, that press the gas. What gas do they press? Ghrelin stimulates the neuropeptide gamma secreting neurons, and then the neuropeptide gamma secreting neurons, they cause hunger. The other types of the neurons are the melanocortin secreting neurons, and then they cause satiety. So we have 2 computers. This is really important. We have 2 computers. The first one is melanocortin, and the second one is neuropeptide gamma. The first one is, the first computer is and then there's a second computer, and they both are antagonists. They work against each other. The first one is melanocortin, and the second one is neuropeptide gamma. What happens is, whenever neuropeptide gamma is stimulated, it leads to hunger. And melanocortin, whenever it's stimulated, it leads to satiety. So the neuropeptide gammas, they lead to increasing hunger and a depressed, and an increased and a depressed appetite. While the melanocortin center, whenever stimulated, it leads to depressed hunger and increased satiety. Sorry, it's supposed to be satiety. So they are antagonists. So whenever neuropeptide gamma is stimulated, that leads to a sensation of hunger. And whenever melanocortin secreting neurons are stimulated, that leads to satiety. Now, who stimulates who? Ghrelin, the secretion of ghrelin, leads to the stimulation of neuropeptide gamma, and that leads to hunger. So ghrelin, stimulation of ghrelin, so ghrelin would stimulate neuropeptide gamma, and then neuropeptide gamma would make you feel hungry and less full. And that would lead you to the intake of the food. Not only that, the polypeptide gamma secreted from the small intestine would inhibit neuropeptide gamma. Would inhibit neuropeptide gamma, and that would inhibit for us hunger, and that would initiate for us satiety. Who else is inhibiting neuropeptide gamma? It's also cholecystokinin and insulin. And cholecystokinin. So these three enzymes work together to suppress hunger and to initiate satiety by blocking our neuropeptide gamma computer, and that automatically leads to a drop in hunger and an initiation of satiety. While the other computer only works with one system, and that is leptin. So the melanocortin secreting neurons, they receive stimulation from leptin that leads to satiety. So this upper computer receives stimulation from leptin, and upon stimulation from leptin, that leads to you feeling a drop in the sensation of hunger, and that would initiate satiety. So these guys are antagonists. So these two computers are antagonists. But one of them receives, so the neuropeptide gamma receives stimulation from ghrelin, and once receiving the stimulation from ghrelin, the neuropeptide gamma would lead to an initiation of hunger and a drop in satiety. This system can also receive inhibitory, so this is an example of excitatory input. So this excitatory input, plus, you're pressing on the gas. So this neuropeptide gamma receives excitatory input from ghrelin, and that leads it to cause an initiation of hunger and a drop in satiety. But the neuropeptide gamma can also receive breaks or inhibitory, minus, inhibitory stimulation, known as inhibitory stimulation from protein gamma and insulin and cholecystokinin, and that leads to a depression or inhibition of neuropeptide gamma. That leads to a drop, which is the opposite antagonist effect, a drop in hunger sensation and an initiation of satiety. While the other computer that is mainly responsible of depressing appetite, depressing hunger, and initiating satiety can only receive one type of input, that is excitatory input. And the excitatory input leads to one output. The excitatory input comes from leptin, is integrated in the melanocortin hypothalamus neurons, and that leads to an output that is depressing appetite, depressing hunger, and initiating satiety. While the other center is responsible for initiating appetite, initiating hunger, and dropping satiety. Is that clear? Are we clear? We are clear. So one center receives, which of the two centers receives both stimulatory and inhibitory? Yeah, neuropeptide gamma. Good. Okay. Hunger is also stimulated partly by gastric peristalsis. So mild hunger contractions begin soon after the stomach is empty. And this distension, so the empty stomach, when the stomach is full with food, the food that's in the stomach will press against the walls of the stomach. That stretch will be sensed by stretch receptors, who will send stimulation. They will send stimulation to your hunger centers, and the parasympathetic nervous system is going to start the peristalsis and the mixing contractions, because now we are responding to the food. We are responding to the food that is distending our stomach. We are responding to it by secreting digestive enzymes, initiating peristalsis, and initiating mixing activity to prepare the food for digestion in the intestines. So increasing the intensity over a period of hours, and they do not affect the amount of food consumed. Appetite is briefly satisfied by chewing and swallowing, stomach filling, and lasting satiety depends upon nutrients entering the blood. So the mere act of chewing, has a reflex satisfaction of hunger. So appetite will be depressed if there is a sensation of your jaw moving and munching and breaking down things. Obesity is having a body weight more than 20% above the recommended norm for one's age, sex, and height. So the body mass index is used as an indication of being overweight or obese. So the body mass index is the weight divided by the height squared. So 20 to 25 is the optimal weight for most people. Over 27 is considered overweight, and above 30 is considered obesity. It shortens life expectancy, obesity, and increased risk of atherosclerosis, hypertension, diabetes mellitus, joint pain, kidney stones, gallstones, cancer of the uterus, cancer of the breast, cancer of the prostate, and sleep apnea. The causes of obesity are hereditary and overfeeding in infancy and childhood. So you have to be very careful with babies and how making a connection between being kind to your baby and feeding it more. So do not have that faulty connection of the more I feed my baby and the more more food, the more ice cream I give to the baby will be, the output is a happy baby. If you go to sleep early, I'll give you ice cream. And then you start to reinforce your baby by getting the outcome that you want and giving them an input that is detrimental to their health in the future. So you have to be very moderate with how you regulate the food habits of the baby from infancy. That's why there's a lot of dietitians and nutritionists that are always involved with pediatricians to help moms, to help train moms how to regulate the food habits of the baby. So do not rely on your mom to help you how to feed your baby. Go to the dietitian and the nutritionist. They will help you based on science, not based on superstition. Okay, so these are MRI images of persons with obesity and normal weight. So if I have an image like this on the exam, which of the two images do you think is an image for a person with normal weight, and which one is an image of an MRI of a person with obesity? Is a tough one to tell. All right, calories. Calories are a measure of the capacity to do biological work. So 1 calorie is the amount of heat required to raise temperature of 1 gram of water by 1 degree Celsius. So a calorie is the amount of heat that is required to raise the temperature of 1 gram of water by 1 degree Celsius. That's a calorie. So 1,000 calories is a kilocalorie, kcal in physiology, or calorie with a capital C in dietitians. So if you spell a calorie with a capital C, that means it's actually 1,000 calories. That's how they trick you on that, the soda and the stuff they put. It's 100 calories with a capital C. It's actually 100,000. But they only put that capital C in there because you would assume that it's just 1,000 calories. But if it with a capital C, it's actually 1,000. So 100,000 calories. Carbohydrates and proteins yield about 4 kilocalories per gram. That's a lot. So per 1 gram, you have about 4,000 calories. Only 1 gram, 4,000 calories. That is a lot. Sugar and alcohol, about 7.1 kilocalories per gram, are known as empty calories. They provide few nutrition and suppress appetite. So you can have one meal that is 1,000 calories full of with a little bit of sugar, but if you have 1,000 calories worth of vegetables, they would provide you with a lot more nutrition, a lot more energy, a lot less stress, a lot less metabolic stress than having 1,000 calories of sugar that would quickly suppress your appetite, but actually input a lot of calorie stress. And fats yield about 9 kilocalories per gram. So that shows you that fats are the highest source of calories, about 9 kilocalories per gram. Nutrients are any ingested chemical used for growth, repair, or maintenance of the body. So there are 6 classes of nutrients. There's water, carbohydrates, proteins, and lipids. Do you see water in there? So on the exam, when you see, is water considered a type of a nutrient? Yes, it's one of the 6. It's water, carbohydrates, lipids, and proteins. Considered macronutrients. They must be consumed in relatively large quantities. Vitamins and minerals. These are micronutrients, and only small quantities are required. So there is a DRI, Dietary Reference Intake. This is a broad system of nutritional recommendations that include recommended daily allowances. These are a safe estimate of daily intake that would meet the nutritional needs of most healthy people. There is adequate intake, that is the used intake of recommended daily allowances if less certainty established for needs. There's essential nutrients. They cannot be synthesized in the body. these are that they must be ingested. They must be taken from outside because they cannot be synthesized in the body. These are minerals, most vitamins, and there are 8 amino acids known as essential amino acids, and 1 to 3 of the fatty acids must be consumed in diet. Now let's begin with carbohydrates. The functions of sugars. They are structural components of other molecules, including the nucleic acids. So the nucleic acid has a ribose sugar. And what is a ATP comprised of? It is comprised of a ribose sugar with a phosphate group. So there's a sugar in there, and connected to either adenine, thymine, guanine, or cytosine. That's the basic structure of a nucleic acid. It must have a nucleotide plus a sugar plus a phosphate group. In ATP, what happens to be the nucleotide? It's adenine. So it's adenine triphosphate, 3 of the phosphates. PPP, 3 phosphates. 3 phosphates, sugar, and adenine. That's what makes it adenine triphosphate. But don't forget the sugar, even though it's not mentioned, but it's there. Adenine triphosphate. Okay. It's glycoproteins. These are proteins that have a covalent association with sugars. There are glycolipids, and these are lipids that have associations with proteins. Lipids that are associated with sugars, sorry. And then ATP and related nucleotides, like GTP. Now, GTP is guanine triphosphate. So now we still have our ribose sugar. And we still have our 3 phosphate groups, but our nucleotide is no longer adenine. What is it now? It's guanine. That's what makes it guanine triphosphate. Most of the sugars serve as fuel. They are easily oxidized. So they are easily oxidized source of chemical energy. Most cells meet the energy needs by a combination of carbohydrates and fats. So neurons and erythrocytes depend solely on carbohydrates. That's why in starvation mode, what happens is all the cells will stop their carbohydrate metabolism and shift all the sugar to the brain. They give preference to the brain when there is deficiency of carbohydrates in the blood, when there is not sugar enough for everybody, when there is hypoglycemia. Carbohydrate metabolism is shut down everywhere in the body except your brain, because your brain relies solely on carbohydrate metabolism. So everybody will give preference to the brain, and carbohydrate metabolism will shut down everywhere in the body, and it will all the sugar will be shunted and shuttled to your brain. Plasma glucose concentration is carefully regulated by insulin and glucagon. So there's a seesaw relationship between insulin and glucagon, and that is to regulate that there is euglycemia. We don't need hyperglycemia or hypoglycemia. We need euglycemia. It's very important to know how is insulin and glucagon secreted. So we need to. This is the pancreas. Is our computer for now. This pancreas will receive input. If there is an increase, is hyperglycemia. The pancreas will receive input of hyperglycemia. There is an increase in sugar in our blood. What happens is afferent neurons will send sensory input stimulation to our pancreas. The pancreas will integrate that there is an increase in sugar in the blood stream, known as hyperglycemia. Then there will be an output response as the secretion of insulin. And then insulin will cause the synthesis of many sugar channels that will be plucked in the membrane of the cells that lead to the uptake of glucose from the bloodstream into the cell, from the extracellular fluid into the extracellular fluid is divided into intravascular compartment and extravascular compartment is actually the intravascular compartment of the extracellular fluid. The sugar will be taken from there into the intracellular fluid, and that will drop the amount of sugar extracellularly, and that will drop the hyperglycemia into the euglycemia range. Then there will be a detection that will apply brakes and inhibition in our pancreas to stop the secretion of insulin. Otherwise, we're now going to move from euglycemia down to hypoglycemia. The other type of stressful stimuli is hypoglycemia. And hypoglycemia represents an input sensory afferent stimulation into our pancreas, and the pancreas will secrete glucagon. And glucagon is going to cause a breakdown of the sugar polymer known as glycogen stored in the liver and the muscles. And that will start plucking off all of these glucoses and sending them, dephosphorylating them. Remember, we talked about how inside the liver, to tag glucose so that it doesn't leak out, we put a phosphate minus group. So now it is now tied. You now you tie it with something with the P minus, the phosphate group. Glucagon, when you secrete glucagon, it will dephosphorylate. Remove that P minus, and now we can easily liberate glucose that will easily flow out of the carbohydrate stores known as the skeletal muscles mainly. Where is sugar glycogen stored more? In the liver or skeletal muscle tissue? Please think. What is bigger, your liver or all the skeletal muscle tissue in your body? So who has a lot of glycogen storage? Your liver or your skeletal muscles? Skeletal muscles. Good. Okay. Okay. Carbohydrates. Daily requirements. So carbohydrates are oxidized rapidly. So are required in greater amounts than any other nutrients. So the reason why carbohydrates are required in greater amounts than other nutrients is that because they are oxidized rapidly. And that formula is on the exam. I put it on the study guide. So you have to be able to tell that 6-carbon glucose will be oxidized with 6 molecules of water, and that with 6 molecules of oxygen, and that will give us 6 water molecules and 6 carbon dioxide. That is the formula for the oxidation of carbohydrates, and it happens so rapidly. It is not a slow process. So the recommended per day is about 130 grams of sugar. The brain alone consumes about 120 grams of glucose per day. And dietary carbohydrates come in 3 main forms: monosaccharides, glucose, galactose, fructose, disaccharides, sucrose, lactose, and maltose, and polysaccharides. Only significant one is starch. This is very important. And this is one of the questions of the short answer. So you must remember that the most significant polymer of carbohydrates is starch. The other one that the animals use is known as what? Cellulose. Cellulose. So cellulose and starch are the biggest polymers, of carbohydrates in existence. So how come animals are able to digest starch and we are unable to digest starch? The animals are able to digest starch because they have enzymes that hydrolyze the alpha 1 4 linkage that is utilized by cellulose. So cellulose is a lot of carbohydrates that are attached to each other by an alpha 1 4 linkage. But starch is actually a beta 1 4 linkage that binds, this beta 1 4 linkage binds all of our starch. So starch has a lot of beta 1 4 linkage, and cellulose has alpha 1 4 linkage. We don't have this enzyme. So when you eat cellulose, you can never break these bonds. But the cow can. The cow has the alpha 1 4 linkage glycosidase. It can break down this bond. So on the short answer, what is the main polymer? What is the main